We'll do a different episode later. Okay. We're not a window, a tab. Cancel. Okay, so we did all of that. We got through all of that. We got to the codon table. I think, or we just finished talking about, or I just introduced the idea of transfer RNA. So we're around here. So we'll just flip through these. We've seen this many times. So the bit that's new basically is this truck that's driving in and out, right? It's carrying the amino acids to the ribosomes. So that's what transfer RNA is basically doing. It's bringing the appropriate amino acid into the ribosome so that we can actually build this protein, which will build your body, right? And that's translation. The T in tRNA stands for transfer RNA, but you could just as easily call it the translation RNA, because what it literally does is it translates from the language of nucleic acids to the language of amino acids. Okay, so this is the translation device. So if you looked at transfer RNA as a like you looked at the RNA structure of it flattened out, it looks basically like a three-leaf clover. So it has three loops in it. Okay. At one end will be the site where the amino acids are attached. And at the bottom end of the clover leaf is the anticodon. These are the bases that are complementary to the codon in the mRNA. So if the anticodon is AAG, then the mRNA would have said UUC. Okay. Now, the structure isn't actually like this in 3 dimensions. It's folded and curved. So it makes sort of this backward 7 shape. Okay. So at one end, you're going to have the anticodons, right? So this is called the anticodon loop when you look at it in this leaf structure. But at the bottom of the structure are the anticodons. And then at the top ends are where the amino acids attach. Okay, so this is where the correct amino acid is going to be attached to. And that attachment is done by this particular enzyme. This enzyme is called, this purple activating enzyme is called aminoacyl tRNA synthetase, because what it does is it synthesizes the link between an amino acid and the transfer RNA. So this is the enzyme which bonds the amino acid, in this case TRP, to transfer RNA. So the transfer RNA is held in place. You notice that there's a hydroxide here and a hydroxide here, which means you can do a hydrolysis reaction, right? So you can remove water, or not hydrolysis, condensation. So you can remove water here. When you do, you're going to form a bond here. Since this is a synthesis reaction, it requires energy. So you're going to use ATP. However, we're going to use it slightly differently. We're going to use up both phosphates. So the ATP is going to go to AMP. Okay. This is a relatively unstable bond. Okay. It's one that can be broken relatively easily. We want that because at the ribosome, we don't want this bond to break so that the amino acid could be attached to the growing peptide. Okay. So that's how we load the empty tRNA with an amino acid. Okay. The other part that's important when we talk about protein synthesis is the ribosomes. Okay, so the ribosomes facilitate or help in the coupling of the transfer RNA anticodon to the mRNA codons. Okay, it's simultaneously an organelle and an enzyme because it does do synthesis. Okay, Structurally, it's made up of two components: the rRNA, ribosomal RNA, and proteins. Okay, and these things combine as two subunits. They're called the large subunit and the small subunit. Okay, when they come together, you have a functional ribosome. Okay, so how do ribosomes work? The way that ribosomes actually allow translation to occur is in three steps. Step number one, well, I should describe the sites on the ribosome first. The first site is where the amino acids are going to come into the ribosome so that they can leave their amino acid behind. Okay, this is called the A site, the amino acyl site, or the amino acid site. Okay, this is where the tRNA carrying the next amino acid to attach is going to be added to the chain. That's this location right here. The next site is called the P site. It's the peptide or peptidal. I prefer site. This is where you're going to have the peptide chain growing. So you're going to have the peptide polypeptide growing in this middle location. And the last site is called the E site for exit. The empty transfer RNA, once the amino acid has been transferred to the polypeptide, the empty transfer RNA has to get kicked out. Okay, so the empty tRNA is going to leave the ribosome from that exit site. Okay, so the A site is here, the P site is in the middle, and the E site is the one on the left. Okay, note that when this system runs, it's the 5' end of the messenger RNA that goes into the ribosome first. Okay, it's like if you've taken in physics, do they 11 and 12? Do they still do ticker tape labs? Okay, so consider the ribosome to be the mechanism and the mRNA to be the ticker tape. Okay, so uh, that's probably the simplest equivalent that I can think of. Okay, so how do we actually go about building the polypeptide? Okay, so there are three steps. Step number one, initiation, right? So in initiation, you got to bring all the different components together. Okay, so you're going to bring together the messenger RNA, the ribosomal subunits, large and small, and the initiator transfer RNA. The initiator transfer RNA is the transfer RNA that's carrying the first amino acid, which is usually methionine. Okay, so here's the transfer RNA carrying methionine. It's There's the messenger RNA, and the messenger RNA has the start codon AUG. Because the start codon is AUG, the anticodon on the transfer RNA has to be the complement UAC. Okay. Because this is initiation, this is the only time that the entering amino acid comes in on the P site. Okay, so only the very first amino acid in the construction of a polypeptide will enter the P site of the ribosome. All other transfer RNAs will enter via the A site. Okay, so the P site reads AUG, brings in the correct tRNA with methionine in it. Okay, you then have elongation. The next in the A site will be another codon, the second amino acid that has to go in here. The codon says CUG, which means the anticodon has to be GAC. So that's going to come into this position. Okay, so the P site has the initial transfer RNA and amino acid. The A site has the second one in position, LEU. Okay. In order to form a polypeptide, what kind of bond has to be formed between the two amino acids? Yeah. So the ribosome will now catalyze a peptide bond to be formed between the first amino acid and the second amino acid, transferring the first amino acid onto the second one, at the same time releasing the transfer RNA. Okay, at this point, the P site is filled and the A site is filled. The only way you can bring in a third amino acid is by reading the next codon. Okay, so what has to happen is that the entire ribosome moves over one complete codon. It shifts. Okay, and when it does so, the empty transfer RNA gets moved into the E site where it will exit. The growing polypeptide is now held in the P site, and the A site is now available for the next amino acid to come in. Since it's AAU, what's going to be brought in here will be the anticodon, which is going to be UUA. So the correct amino acid for UUA, let me just find the codon table. So for UUA, that's leucine. Okay, so what's going to happen is the amino transfer RNA with another leucine is going to come in, attach here, and then this will again repeat. New peptide bond will form. The entire system will shift over one codon, and the fourth codon will be opened up. Okay, so this shifting is going to continue all the way down until we get to termination. Okay, at termination, you're going to read a stop codon, in this case UAA. When UAA is read by the ribosome A site, a release factor is going to be brought in. When that release factor comes in, it causes the final bond here to break. It causes the small and the large subunits to separate out. And it causes the protein itself to be released. Okay? The two subunits will then head back to the beginning of the mRNA, reattach at the AUG start codon, and start making another protein. And this will continue for as long as the mRNA doesn't degrade. Okay? That's the entire sequence. Okay? Before you can ignore this protein signaling thing. Before I talk about that slide, so slide 36, just cross off and ignore. You don't need to worry about it. Okay. But what I do want to show you next is I'm going to show you this particular animation on YouTube. It loads.
With MailChimp's customer journey smarts, you can track when a customer buys something, plan for their next purchase.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of
It's just a protein that's going to cause the system to fall apart.
the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay, so that I have put that slide onto the classroom as well, so you can review translation there using that video. Ignore this slide, some number 36. We're not going to talk about it. Okay, so the idea is, can you describe the entire process, starting with DNA and RNA polymerase and ending up with the final polypeptide, right? So in terms of the components that you should be able to talk about, there's the DNA, the RNA polymerase, the primary transcript, exons, introns, the 5' and cap, and the 3' poly A tail, the mature RNA transcript. You should also be able to talk about spliceozymes here, spliceosomes, the large and the small ribosomal subunits, okay, coming together to form a ribosome, the 3 sites on the ribosome, the ends of the ribosome of the messenger RNA and how it enters the ribosome, what a transfer RNA is, how the empty tRNAs and amino acids get joined by aminoacyl tRNA synthetase, okay, and then the formation of the polypeptide, okay. So what we've been talking about for the past 2 days or so is summarized completely in this diagram. So you should be able to tell me the entire story from here, okay. If you can't, go back and review your notes, okay. What's really hard now at this point then is, what the heck is a gene? What is a transcriptional unit? Okay. Is it this bit here, right, on the DNA that gets completely translated? Does it include the introns? Okay. Or is it just this section? Is it what we call the pre-mRNA? Or is it the mature RNA? What exactly is a gene? And what are we actually manipulating when we talk about genetic engineering then? Okay, so that's a question that no one's really ever settled on. Okay, um, depending on what aspect of genetics, molecular genetics you work on, you may have a different definition of what a gene is from a colleague. Okay. All right. One of the things I want to talk about next is the differences between protein synthesis between eukaryotes and prokaryotes. Okay. And as as Fillmore says, one of the biggest differences between the two systems is no nucleus. Okay. So if we look at the genomes of prokaryotes and eukaryotes, okay, in prokaryotes, the DNA is in the cytoplasm. In eukaryotes, it's located within the nucleus. Prokaryotes have circular chromosomes. Eukaryotes have linear chromosomes. Okay, prokaryotes have just DNA. Eukaryotes have DNA plus proteins. Prokaryotes, their genes are continuous. Okay, no introns. Whereas eukaryotes have introns and exons. Okay, which makes them a little bit more complicated to deal with. Okay, remember that the introns come out. So this is what's actually quite interesting. I know this picture here is a little bit hard to figure out, so we've redrawn it down here. Okay, the light, thin, purpley colored line here, this is prokaryotic DNA. Because they couldn't tell where the protein was coming from. We now know that the DNA in prokaryotes is naked, but back then, because of the way they were doing their extractions, they were getting a mixture of proteins and DNA. Okay, the light pink dot here is DNA polymerase. Okay, so what must these vertical strands or almost semi-perpendicular strands coming off the DNA, what must they be? They're not peptide chains yet because they have ribosomes on them. Yeah, that's the purple dots. I'm talking about the very thin blue line running through the ribosomes. That's the messenger RNA, right? As you see down here. Okay, and then you've got the ribosomes up top on top of it, these purple blobs. Okay, this situation would never occur in a eukaryote. Why not? Yeah. So what's neat about prokaryotes is that in prokaryotes, transcription and translation are simultaneous. Because the nucleus is because the genetic material is in the cytoplasm and there isn't a nuclear membrane and there's no introns to get rid of, you can have translation and transcription occur at the exact same time. So each one of these purple dots is translating at the same time the RNA polymerase is transcribing the messenger RNA. Okay? Which makes this system a heck of a lot faster than what occurs in us. Yeah, it does. Because remember, the other reason that prokaryotes get away with it, prokaryotic DNA is circular. It doesn't have an end, so there's nowhere for an enzyme to start attacking. Our DNA is linear. Since it's linear, there's an end for the enzymes to start digesting the DNA. So to protect it, we need it inside the nucleus. There are other reasons as well, compartmentalization and things like that. But at this level, yeah, the protection is probably the primary factor. Okay, so differences between prokaryotes and eukaryotes, the length of time and the physical separation between the processes. Eukaryotes takes about an hour from DNA to get the protein. And then in prokaryotes, there is no RNA processing. So it's a lot faster in these guys. Okay. All right. So the next thing I want you guys to watch is the crash course on protein synthesis. Okay, it is long. It's 15 minutes or so. Uh, and it's a pretty good review because it goes into everything we've been talking about and also reviews protein structure, primary, secondary, tertiary, quaternary.
Ugh, I'm so done with this. What's wrong? For the past 3 hours, I've been held up by useless information
RNA. The ribosome is also bringing in whatever amino acid.
Hang on. Start it from the
Okay,
beginning.
roll it. You know what this is? It is the longest word in the world. Like anywhere, any language, more than 189,000 letters. If you were to write it down, though I don't know why you would, it would fill up more than 100 pages, and if you could actually say it without like breaking your face, it'd take about 5 hours. What the frick is this word? Well, it is the name of the longest known protein on Earth, and it's actually in you right now. Because of its enormous size, it was given the name Titan by scientists, and that's with 2 I's. And it's protein that helps give some like the springiness to your muscles. Today we're going to be talking about DNA and how it, along with 3 versions of its cousin RNA, unleash chemical kung fu to synthesize proteins just like this. So this is going to take a while to explain. So how about we make ourselves some Hot Pockets? Those are my favorite, ham and cheese. Every time I take a bite, I wonder, how do they do it? How do they pack exactly the same flavor into every foil cardboard wrapped food-ish item? Clearly there's got to be some super secret instruction manual kept in a location known only to 2 people. And since I'm talking about
Scientists,
biology
apparently.
here, that brings up a related question. How did I get built from the DNA instructions and biological molecules we've been talking about? Today, that's what I'm going to do. Not actually make Hot Pockets or a person, but I'm going to be talking about DNA transcription and translation, which is how we get made into the delicious things that we are today. Though, hopefully none of us know how delicious people are. Animals, plants, and also Hot Pockets really are nothing more than salty water, carbohydrates, fats, and proteins combined in precise proportions following very explicit instructions. Let's say I want to make my own Hot Pocket. I would have to, 1, break into the lair of the Hot Pocket company holding the secret manual. 2, read the instructions on how to make the machinery to produce the Hot Pocket and the proportions of the ingredients. 3, quickly write down that information in shorthand before I get caught by the Hot Pocket police. 4, go home, follow the instructions, build the machinery, and mix the ingredients together until I have a perfect Hot Pocket. And that is how we get us. Very simply, inside the cell's nucleus, the DNA instruction manual was copied gene by transcription onto a kind of RNA and then taken out of the layer where the instructions are followed by the process of translation to assemble amino acid strings into polypeptides or proteins that make up all kinds of stuff from this Titan down here to the keratin in my hair. But most of the polypeptides that get made aren't structural proteins like hair, they're enzymes which go on to act like the assembly machinery, breaking down and building and combining carbohydrates and lipids and proteins that make up variations of cell material. So enzymes are just like whatever ingenious machinery they use the factory to make this. Okay, let's start out in the layer. The nucleus. The length of DNA that we're going to be transcribing onto an RNA molecule is called our transcription unit. And let's say, in today's example, that it's going to include the gene that transcribes for our friend Titan, which in humans, at least, occurs on chromosome 2. Now, each transcription unit has a sequence just above it on the strand, and that's called upstream. Biologists call that upstream on the strand, and that sequence sort of defines when the transcription unit is going to begin. This special sequence is the promoter, and it almost always contains a sequence of 2 of the 4 nitrogenous bases that we talked about in our last episode: adenine, thymine, cytosine, and guanine. Specifically, the promoter is a really simple repetition. We got thymine, adenine, thymine, adenine, and then A, and then on the other side, AT. Because you know how this works, right? This is called the ta-ta box. It's nearly universal, and it helps our enzyme figure out where to bind to the strand. Now you'll remember from our episode about DNA structure that DNA strands run in one of 2 directions, depending on which end of the strand is free and which end has a phosphate bond. One direction is 5' to 3', and the other direction is 3' to 5'. In this case, upstream means toward the 3' end, and downstream means toward the 5' end. So the first enzyme in this process is RNA polymerase, and it copies the DNA sequence downstream of the ta-ta box. That's towards the 5' end, and copies it into a similar type of language, messenger RNA. Quick aside, so you'll notice that to read the DNA in order to make enzymes, we need an enzyme in the first place. So it kind of gets chicken egg here. We need the enzyme to make the DNA and the DNA to make the enzyme. So where did RNA polymerase come from in the first place if we haven't made it yet? What an excellent question. It turns out that all of these basic necessities get handed down from your mom, she packed quite a lot more into her egg than just her DNA, so, we had a healthy start. So, thanks, Mom. So the RNA polymerase binds to the DNA at that ta-ta box and begins to unzip the double helix. Working along the DNA chain, the enzyme reads the nitrogenous bases, those are the letters, and helps the RNA version of those nitrogenous bases floating around in the nucleus to find their match. Now, you might also recall from our previous episodes that nitrogenous bases only have one counterpart that they can bond with, but RNA, which is the pink one here, doesn't have thymine like DNA does, which is the green and the blue. Instead, it has uracil. So U appears here in T's place as a partner to adenine. As it moves, the RNA polymerase rezips the DNA behind it and lets our new strand of messenger RNA peel away. Eventually, the RNA polymerase reaches another sequence downstream called a termination signal that triggers it to pull off. Now, some finishing touches before this info can safely leave the lair. First, a special type of guanine is added to the 5' end. That's the first part of the mRNA that we copied, and that's called the 5' cap. On the other end, it looks like I fell asleep with my finger on the A key of my keyboard, but another enzyme added about 250 adenines onto the 3' end. This is called our poly A tail. These caps on either end of the mRNA package make it easier for the mRNA to leave the nucleus. They also help protect it from degradations from nearby passing enzymes, while also making it easier to connect with other organelles later on. But that's still not the end of it. As if to try to confuse me to protect the secret Hot Pocket recipe, the original recipe book also contains lots of extra misleading information. So just before leaving the nucleus, that extra information gets cut out of the RNA in a process called RNA splicing. And it's something like editing this video. This process is really complicated, but I just had to tell you about two of the key players because they have such cool names. One, the SNRPs, which are small nuclear ribonucleoproteins. These are a combination of RNA and proteins, and they recognize the sequences that signal the start and end of the areas to be spliced. SNRPs bunch together with a bunch of other proteins to form the spliceosome, which is what does the actual editing, as it were, breaking the junk segments down so their nitrogenous bases can be reused in DNA or RNA, and sticking together the two ends of the good stuff. That good stuff that gets spliced together, by the way, are called the exons because they will eventually be expressed. The junk that gets cut out are just the intervening segments or the introns. The material in the introns will stay in the nucleus and get recycled. So, for instance, Titan down there is thought to have hundreds of exons when it's all said and done, probably more than 360, which may be more than any other protein. And it also contains the longest intron in humans, some 17,000 base pairs long. Man, Titan, it is just a world record holder. So now that it's been protected and refined, the messenger RNA can now move out of the nucleus. Okay, so a quick review of our Hot Pocket Mission Impossible caper so far. We broke into the layer containing the instructions, we copied down those instructions in shorthand, we added some protective coatings, and then we cut out some extra notes that we didn't need, and then we escaped back out of the lair. Now I have to actually read the notes, make the machinery, and assemble the ingredients. This process is called translation. So next, rewind your memory or just watch that video again to the episode about animal cells. Do you remember the rough endoplasmic reticulum? I hope you do. Those little dots on the membranes are the ribosomes, and the processed messenger RNA gets fed into a ribosome like a dollar bill into a vending machine. Ribosomes are a mixture of protein and a second kind of RNA called ribosomal RNA, or rRNA. And they act together as a sort of workspace. rRNA doesn't contribute any genetic information to the process. Instead, it has binding sites that allow the incoming mRNA to interact with another special type of RNA, the third in this caper, called transfer RNA, or tRNA. And tRNA really might as well be called translation RNA because that's what it does. It translates from the language of nucleotides into the language of amino acids and proteins. On one end of the tRNA is an amino acid. On the other end is a specific sequence of 3 nitrogenous bases. These two ends are kind of matched to each other. Each of the 20 amino acids that we have in our body has its own sequence at the end. So if the tRNA has the amino acid methionine on one end, for instance, it can have UAC as the nucleotide sequence on the other end. Now it's just like building a puzzle. The mRNA slides through the ribosome reads the mRNA 3 letters at a time, each set called a triplet codon. The ribosome then finds the matching piece of the puzzle, a tRNA with 3 bases that will pair with the codon sequence. That end of the tRNA, by the way.
Anyone notice the mistake they've made here in this step? Yeah, so this methionine is actually in the wrong site. It should really be here in the P site instead of being in the A site.
Way is called the anticodon. Sorry for all the terminology. You need to know it! And of course, by bringing in the matching tRNA, the ribosome is also bringing in whatever amino acid is on that tRNA. Okay, so starting at the 5' end of the mRNA that's fed into the ribosome, after the 5' cap, for almost every gene, you find the nucleotide sequence AUG on the mRNA. The ribosome finds a tRNA with the anticodon UAC, and on the other end of that tRNA is methionine. The mRNA, like a mile-long dollar bill, keeps sliding into the ribosome so that the next codon can be read, and another tRNA molecule with the right anticodon binds on. If the codon is UUA, then the matching tRNA has an AAU on one end and a leucine on the other. And if the mRNA has an AGA, then the matching tRNA has a UCU on one end and an arginine on the other. In each case, that new amino acid gets connected onto the previous amino acid, starting a polypeptide chain, which is the beginning, the very beginning of a protein. But it turns out that there are lots of different ways to read this code, because UUA is not the only triplet that codes for leucine. UUG does too, and arginine is coded for by 6 different triplets. This is actually a good thing. It means we can make a few errors in copying and transcribing and translating DNA, and we won't necessarily change the end product. The process continues, with the mRNA sliding in a bit more, and the ribosome bringing in another tRNA with another amino acid, and that amino acid binding to the existing chain, and on and on and on and on and on. Sometimes for thousands of amino acids to make a single polypeptide chain, for example. This whole word is basically just the names of the amino acids in the sequence and the order in which they occur in the protein, all 34,350 of them. But before we can make our own Hot Pockets, and that string of amino acids becomes my muscle tissue, we have some folding to do. That's because proteins, in addition to being hella big, can also contort into very complex and downright lovely formations. One key to understanding how a protein works is to understand how it folds, and scientists have been working for decades on computer programs to try and figure out protein folding. Now, the actual sequence of amino acids in a polypeptide, what you see scrolling along down there, is called its primary structure. One amino acid covalently bonded to another, and that one to another, and to another in a single file. But some amino acids don't like to just hold hands with 2 others. They're a bit more promiscuous than that. The hydrogens on the main backbone of the amino acids like to sometimes form bonds on the side, hydrogen bonds, to the oxygens on amino acids a few doors down. When they do that, depending on the primary structure, they bend and fold and twist into a chain of spirals called a helix. We sometimes also find several kink strands laying parallel to one another called pleated sheets. All those hydrogen bonds and pleated sheets are what make silk strong, for instance. So in the end, our promiscuous amino acids lead to wrinkled sheets. These hydrogen bonds are what help give these polypeptides their secondary structure. But it doesn't end there. Remember the R groups that define each amino acid? Well, some of them are hydrophobic. And since the protein is in the cell, which is mostly water, all of those hydrophobic groups try to hide from the water by huddling together. And that can bend up the chain some more. Other R groups are hydrophilic, which, if nothing else, means that they like to form hydrogen bonds with other hydrophilic R groups. So we get more bonding and more bending, and our single file line has now taken on a massively complex 3-dimensional shape. It also explains why I can fix my bed head by wetting my hair with water. The water helps break some of those hydrogen bonds in the keratin, which relaxes its structure. That way I can comb it out, and when it dries, those bonds reform, and voila, perfect hair. All of this shape caused by bonding between R groups gives our polypeptide its tertiary structure. So now we have a massively contorted polypeptide chain, and it actually contorts very precisely. Sometimes just one chain is what makes up the whole enzyme or protein. In other proteins, like hemoglobin, several different chains come together to form a quaternary structure. So quick review of structure. The sequence is the primary structure. The backbone of hydrogen bonds forming sheets and spirals are the secondary structure. R group bonds are tertiary, and the arrangement of multiple proteins together give the quaternary structure. These polypeptides are either structural proteins, like this thing at the bottom here, that you can find in your muscle, or in my Hot Pocket. They might also be enzymes. Enzymes like do stuff. They can cut up biological molecules like I do with the chef's knife. They can mix stuff, and they can put stuff together. So from that one recipe book, we get all the ingredients and all the tools necessary to make me, which is better than a Hot Pocket. Would you all agree? Now take your time with this stuff. Feel free to watch the episode a couple of times, because next week we're going to talk about how cells swap all of this genetic information through reproduction. Thank you for watching this episode. By now, you should probably know how this works. You can click on any of the links over there, and it'll take you back to that point in the show, as long as you're not watching on your cell phone. It doesn't work on cell phones. I apologize for that. Thank you to everyone who helped us put this show together, and thank you to you for watching it today. If you have any questions about this episode, please leave them in the comments below, or you can get us on Facebook or Twitter. And that's all. Goodbye.
just to show you something, because Abby was talking about it. Here's the full chemical name for Titan. Starts here. It is, let's see, in terms of length, it has 189,819 letters. And here's where it begins. Okay, so it's long. Here, it begins here. Not even going to bother trying to pronounce it. You can see why it was scrolling by so fast in the Hank Green video. Getting there. Yeah, I'll be giving you your gizmo today. I gave it to the other class this morning. Yeah, so it is long. Although there's some debate if it's really a word, right? Some people say that it's a verbal formula rather than an English word. I wonder what the longest word is. So none of these are actual words you would actually use. Well, the other one you've probably heard of is probably supercalifragilisticexpialidocious, if you've watched Mary Poppins. Sure. The largest non-contrived and non-technical word. Yeah, I can see that. 28 letters. Imagine if you tried to put that word for Titan into a dictionary. Okay. Um, you guys are B block. All right, so the next thing I need to take a look at. Okay, we need to do the mutations bit. Okay, I'm gonna say take a 10-minute break right now. It's 1:21. Let's come back together at 1:35, and we'll talk about mutations. Then I'll talk about the study guide and the gizmo. Probably have a work period coming up tomorrow. Well, we have to do operons, which is Wednesday. Thursday is going to be a gizmo day to work on the protein synthesis gizmo for this unit. Okay, so I'll see you at 1:35. Okay. One of the other things I want to talk about, I might as well set this up now. We're close to being done this uh, unit. As you can see here on the calendar, for the other class, I've set their genetics test for the 10th. That's what they've asked for. Um, we have one gizmo and on protein synthesis to do. That'll be on Thursday, Wednesday, tomorrow and Thursday. And we have one slide deck left on after today on operant, and we're done. So the question is, when do you want your unit test? Same day, the 10th? Tuesday, the 8th. So Grant says Tuesday. I'm not sure what Vedant is saying sure to. Well, we're not test. Yeah. The 9th is a BFF day, so there will be nothing going on anyways. The Fridays are getting a little late. I really don't want to go any later than Thursday. I can't do it on Wednesday because Wednesday is a BFF day. There are no evaluations or things like that allowed on a BFF day. It's a makeup day, so I can't do the 9th. So how about hands up for Tuesday the 8th? 1, 2, 3. Okay, hands up for Thursday the 10th. Okay, so the people who said the 9th or the 8th, do you have a test on, and that's why, or is it, because I could have you write it, those of the three of you, I could have you write it early. Okay. I think more people want so the extra time to study. So I'm gonna say Thursday the 10th then. So set it for the same date. So that is the block B genetics test. We then. So I'll do the review on the 8th since I can't do it on BFF day. Okay, so that's one thing set. The other thing, I guess I will just add it now. Reuse post is the gizmo on protein synthesis. And you guys are going to have a few days to work on that. You'll have tomorrow and Thursday, so I'm going to make that due on the 7th. And assign. Um, and that will be your last test, the genetics one. There won't be any more tests after that. We'll have papers or projects, but nothing testing wise. I might do like a 5-point quiz thing, multiple choice, but there's not going to be any actual tests. Okay. So that's your last test for Bio 12. Okay. Also want to remind you tomorrow is another one of those one-on-one sessions. So we have 3 of those left. So we'll start with that tomorrow first thing. And in terms of finishing off today, we're going to look at this, which is mutations. It's fast. I think it's only like 9 slides. So this is not going to take too long. Okay. So let's open that up. Okay. So what's a mutation? What is the definition of a mutation? Okay. It's even simpler than that. It's a change in genetic sequence. Okay. Now, Vedant said something that a lot of people often say, which is that it's a change in DNA. Does a change have to be just in the DNA in order for a mutation to occur? Or can a change occur anywhere else? Yeah. So remember that for mutations, mutations can occur in either nucleic acid type, DNA or RNA. Okay. The only difference is that in DNA, it's in the nucleus and it may end up affecting a lot of different cells, whereas in RNA, it may only affect one round of protein synthesis. Okay? That's the real big difference. Okay. So let's look at these mutations. Okay? There are 2 basic kinds of mutations. So the ones we're going to look at are changes in gene sequences in DNA or RNA. And so we call these basically like point mutations. This is different from chromosomal mutations. So last year in grade 11, we would have talked about things like non-disjunction, right? Trisomy 21 Down syndrome, Klinefelter syndrome, XXY, um, excuse me, triple X, all of those different kinds of chromosomal errors. There would also be times when you could have the chromosome either lose a chunk of the chromosome, double up on the chromosome, invert the chromosome. Those kinds of errors are what we call chromosomal mutations. We covered those last year. That's not what we're going to talk about right now. We're talking about mutations that occur to the DNA or RNA sequence that change the genetic message. Okay? So in a point mutation, we're talking about a single base unit change. Okay? Usually these are called base pair substitutions. So up at the top, this is the normal or wild type sequence. AUG, AAG, UUU, GGC, UAA. Now, I'm using an RNA for the example, but it could just as well be DNA. So in a base pair substitution, you're changing a base that is present for a different one, okay? So if you look at the 4th codon, right, in the normal DNA or RNA, it's GGC. But in the first mutation, instead of the C, we substitute it with a U. So the 4th codon goes from a GGC to a GGU. But is there any change in the amino acid that's brought in? No. So GGC codes for glycine and GGU codes for glycine. So the amino acid doesn't change, even though a mutation did occur. Okay, this is a silent mutation. There's no change in the amino acid, even though the nucleic acids did change. That's due to the redundancy in the code and the whole idea of third base wobble. So you can have a mutation occur that has absolutely zero impact. Okay. The second type of mutation that you can get is a missense mutation. If you look at the same fourth codon, you go from a GGC to an AGC. That does change the amino acid present. So we go from GLY to SER. Okay, so from glycine to serine. This may or may not have an impact on the functioning of the protein. It depends. Okay, but this is a missense. So a missense mutation is like a typo when you're word processing. So it's a spelling mistake basically. Okay. The third type of mutation you can get is called a nonsense. Look at the second base. So the second codon is AAG normally. But if that first A mutates to a U, so it becomes a UAG. Well, UAG is a stop codon. So the protein is going to be methionine and then nothing else. So it shortens the um, protein, which is going to render it probably non-functional. Okay, and it doesn't make any sense. So they call this a nonsense mutation. Okay, so base pair substitutions can end up basically with three different kinds of results. Okay, now when will a mutation affect your children? Yeah. So if the mutation occurs in the reproductive cells, then the next generation has to worry. Okay. So if you get a mutation that causes you skin cancer in your epidermal layer, that's upsetting for you, yes, but it has no impact on your offspring because you can't pass it on. But if the DNA that codes for your epidermis is mutated in your gametes or in your reproductive cells, then you will pass it on. So this is what they mean when they say mutation. It depends. If you get skin cancer from too much sunlight, right? Um, that's not going to be hereditary. But if you get breast cancer and you have the gene BRCA1 and BRCA2, those particular genes are in the nucleus of the cell and they will be present in your reproductive cells, in which case it will be hereditary, right? So it depends on where the mutation occurs. That's why breast cancer can be hereditary in both men and women, right? It is possible for men to get breast cancer. It's incredibly rare because men don't have that much breast tissue that can present that can mutate that way, but they have some. So there are cases of men getting breast cancer. Okay, um, but if you get skin cancer because of exposure to something, then no, it's probably not hereditary. Okay, so if we look at something like sickle cell anemia, which is hereditary, it's hereditary because these mutant DNAs are also found in the reproductive cells. That's why it can be passed on. Okay. So in normal hemoglobin, you would have CTT. So the mRNA would code GAA, which gives you the amino acid glue. Okay. But if you have sickle cell anemia, then you received a mutation in your hemoglobin DNA that changed CTT to CAT, giving you an mRNA codon of GUA, which codes for valine. Okay, so what kind of mutation is this of the three that we've been discussing? Silent, missense, or nonsense? Yeah, this is a missense mutation. It's a typo. Okay, why does this cause sickle cell anemia? Okay, well, GLU, amino acid number 6 on the hemoglobin normally is hydrophilic, but VAL is hydrophobic. So it literally changes the secondary structure and the tertiary structure of the hemoglobin molecule, causing it to deform in this manner. Okay. So, and because it occurs in reproductive cells in the gametes, it's a recessive inheritance pattern, and it strikes about the same percentages in African Canadians as well, about 1 out of every 400. Okay. Another recessive genetically inherited, oh, actually, we'll talk about it after. Um, so the other kind of mutation you can get is called a frame shift. Okay. What do you think is happening here? By the name, a frame shift mutation. What do you think has happened in this mutation type? Okay, close. So when they talk about a frame shift, what they're talking about is the reading frame. Okay. And the reading frame is the 3-letter codon that's being read. Exactly, Abby. So the reading frame in the top DNA sequence is AUG, AAG, UUU, UGC, UAA. So the reading frames are broken up into triplets. Okay. But if that one of these bases is either added to or deleted, then from the point where that change occurs to the rest of the enzyme will be affected. So for example, if you have an insertion, okay, so if you look at this one, okay, the second amino acid, the second codon is AAG. If you insert an extra U here, that becomes UAA, and look what happens. The third amino acid, instead of being UUU, because of the addition, becomes AGU, because you've shifted everything over by one nucleotide base. Yes, spliceosome mistakes could cause this. It could also be caused by replication error. It could be caused by mismatching of chromosomes. Yeah, there's a lot of different ways this can happen. Okay, if you look at the first one, if you delete a base, right? So UUU, that U goes missing, then that's going to become UUG, then GCUAA, right? And so you change the sequence from met, lice, phi, glee to met, lice, lu, alla. Okay. So adding bases or deleting bases will cause the reading frame to shift over and therefore completely screw up the sequence of your DNA from that point on. Okay. Where would this cause more damage? If it's near the beginning of a DNA sequence or near the end of a nucleotide sequence? Yeah, more cells are going to be impacted if it's at the beginning. Correct. Okay, so a couple examples, or one example of this, cystic fibrosis. Okay, usually impacts whites of European descent. Okay, so Germany, France, Spain, Italy, the Baltics states, Poland, Hungary, Austria, all of the Switzerland, all those nations. If you have that background, okay, 1 out of every 2,500 live births will have a child with this particular condition, and about 1 out of every 25 Caucasians is going to be a carrier of this condition. Okay. So what happens in this illness is that there's a mutation in the genes that code for a chlorine transporter. Okay. So the chlorine transporter becomes defective. Since it's defective, you limit the amount of chlorine in water that can be transported across the cell membrane. That results in a thicker and stickier mucus coat around all the cells, and that mucus buildup causes problems in the pancreas, lungs, and digestive tract, and causes bacterial infections because the mucus now allows bacteria to land on it and start growing there more easily. So It causes a lot of problems. Without treatment, children will usually die before age 5. With treatment, they can live past their late 20s. Okay, so this is basically the impact, right? The mucus builds up so thick, and you get all these bacteria building up on these mucus secreting glands. And obviously, if this builds up in the respiratory tract, it's going to be really easy to get some kind of lung illness like pneumonia or anything along those lines, which is problematic. And with the mucus buildup, what's it going to do to the diffusion distance for oxygen and CO2 here in the lungs? Yeah, larger, making it harder to breathe. Okay. So what happens in this particular illness is that people who have this condition usually have these three bases, CTT, on chromosome number 7 somehow gets deleted. Okay. And so this mutation is called delta F508. You don't need to know that. I'm just giving that to you for completeness. Okay. So if you have ended up, if you have this deletion in chromosome 7, you end up with um, the cystic fibrosis transfer gene. Okay. That's what they end up calling it. And it causes your chlorine transporters not to work properly. Okay. And so you end up with those conditions. Okay. So it's effectively you lose one amino acid, causes all these problems. Okay. What these mothers are doing, they're basically holding their kids upside down and they're tapping on their chest fairly vigorously. What that does is it helps break the mucus up in their lungs so that the kids can cough all that mucus out, in addition to medications and such, so that they can breathe easier and continue to live. Okay. And this you should know the answer to. Okay. It's 1:59. There's 20 minutes remaining in class, but I don't see much point in working on the operons. We'll do that tomorrow. So what I'm going to suggest is that if you go to My Nelson, uh, oh, My Nelson, not My Nelsob. Log in, open your text. So you probably want to take a look at chapter 7. What we've been looking at, we finished 7.3 translation, which starts on 324. And there are some questions if you want to answer them on page. Come on, turn. 331. Zoom in on them. See which ones you should be doing. Okay, so on page 131, yeah, 1 through 8. And then the other thing to look at is 7.5, genetic mutations, which is what we just looked at. Question on page 345. You can do 1, 2, 5. One, 2, 4, 5, and 8 is fine. Okay. And then on page 130, 3, 4, 5, 1, 2, 4, 5. I think I said 8. One, 2, 4, 5, and 8. So to take a look at those until 220, and then you're dismissed. So you guys, both classes finished up the mutations. Okay, I'll take those up in class tomorrow. If you get them done, great. If not, not a big deal, because this is really for review. Okay, if you need me, I'm here. Otherwise, I'm going to mute myself and turn my camera off.
All right, guys, it's 19 after. You are dismissed. We'll see you tomorrow.
Thank you. Have a good day.
You too.
Bye, thank you.
Take care.